Hey. So Matthew, I'm not sure if you want me to start or if we're still waiting on some people. Just let me know.
Yeah, I'll give people a minute to log in here. I guess I'll just remind people, for those of you who are enrolled in MC 2111. This is one of we'll have about 8 seminars during the semester, roughly every other week. It will be 2 weeks from now. We'll have Paula Lauren speak to us on the 15th. So again, the deliverable will be you are expected to attend the seminars, and of the 8 seminars, you will write a 1 to 2 page report on basically covering what was maybe a paragraph on what the talk was about, maybe a paragraph on what you thought was interesting about it, and maybe a paragraph on what you would like to do with it if you did decide to do a senior project further on in your studies on this particular topic. So that's basically the deliverables, but we're fairly laid back. The big thing is coming in here, enjoying yourselves, and seeing what the math and computer science department at Lawrence Tech is doing. So just let's see, the participation looks like most of the people expected are in here. So I'll just go ahead and introduce our speaker for today's math and computer science seminar. So Bruce Pell joined Lawrence Technological University in 2019 as a tenure track assistant professor in the mathematics and computer science department. Before that, he graduated from Oakland University in 2010 and did a PhD at Arizona State University and graduated in 2016. In between then and before starting at Lawrence Tech, he was a postdoctoral fellow at St. Olaf College in Minnesota. So without any further ado, I turn the floor over to Professor Bruce Pell to tell us about some disease spread modeling and a little bit of the history of disease spread modeling.
All right. Thank you, Matthew. And hello, everyone. So yeah, like Matthew was saying, I did my undergraduate over here in Michigan at Oakland University, and then afterwards I went to Arizona State for my graduate studies. Today I'll be talking to you about some of my, pretty much like my previous research, my current research, and then some future research that I would like to get into. A lot of it, the talk is a little more broader than a typical like seminar talk, just because I wanted to at least give you the overview of the topics that I work on. And so we'll just kind of go through broadly the kind of the topics and not dive too deep into all the details and things like that. Let's see. So a quick outline. Oh, I should probably check. Everyone can see these slides, right? It's working correctly?
Yes.
Okay. Yeah, so real fast, an outline, we'll talk about, well, we already kind of know who I am, and then we'll talk about why specifically disease modeling, why did I kind of pick it and why it matters, at least to me. And then we'll dive into some previous research projects that I've worked on when it comes to disease spread of specifically infectious diseases. And then I'll dive into the more current and future research projects that I'm interested in. So this is me. My hometown is St. Clair Shores. From the area. My office is 121A. If you're interested in perhaps talking to me, you can either swing by my office or send me an email. A lot of my research is like Dr. Johnston and Dr. Nelson. A lot of it has to deal with the mathematics of biology, biological systems, and specifically, I'm really interested in the idea of disease spread and modeling that. But it doesn't need to necessarily be like infectious diseases. It could be the spread of like a tumor, a cancer tumor in the body or something like that. Interested in all of that kind of stuff. So before we get into the actual, like my research topics and previous projects, I wanted to at least talk about why understanding the spread of diseases is important And I thought I'd start with perhaps kind of one of the founders of modern epidemiology. His name is John Snow. Um, and he was a English physician, a leader in anesthesiology and medical hygiene. And he's pretty famous for his work on cholera. And let's see, yeah, so John Snow and cholera. So if you're not aware, cholera, it's an infectious and often fatal bacterial infection. And it's usually contracted by drinking contaminated water. And in England, 1854, there was a cholera outbreak that occurred. And what made John Snow so famous is that he went out and investigated where these cases of cholera were occurring. He made a map and plotted it. And here's kind of like a more modern, updated version of his map. And what he found was that a majority of the cases were clustering near a water pump where people were collecting their drinking water. So what happens is, and here's, I think, yeah, here's a blown up view of that, the modern map. So the red dots, right, are the cases, and the bigger the circle, right, the more cases were occurring there. And you can see like the little symbols that represent the water fountains. And right in the middle, right, there's this water fountain, and a bunch of cholera cases were found to be around it. And so what he did was basically he was able to trace the infection. Where is it occurring, right? What's the source of the infection? And he traced it back to this water pump. And so what he did was he removed the handle from the water pump so that people couldn't collect water from that specific pump. And so that was one of the first situations where like modern epidemiology occurred. And now I'm seeing Jon Snow from Game of Thrones. Yeah, right. Yeah. So there's I was trying to come up with some kind of reference or joke with that, but I couldn't think of any. Thank you. Yeah. Yeah. So here's the solution, right? What he did. And this is in the 1800s, right? He goes back and he takes the handle off so people can't collect water, right? And basically, he helps out this this spread of disease, right? He slows the spread of it. And actually, you can go there today, right? Modern times, and there's this monument of the water pump with the missing handle. You can go there and visit it. There's a placard and everything. So that's just a nice example of kind of well, the first like modern epidemiological study in a way, right? He studied how the disease was spreading, plotted everything, figured out where this source for the disease was coming from. Whoops, let's see here. Okay, putting it into perspective, I've just listed a couple of some infectious diseases here, and kind of just wanted to note like the global burden. Like, because remember, like, why is this important, right? Why, like, where's the data to show that this is actually, right, an important thing to study and understand and try to mitigate this disease spread. So here's just some cholera, smallpox, Black Plague, the 1918 influenza pandemic. Note, right, there's typical like 4 waves of that epidemic. And 20 to 50 million were estimated to die from that pandemic. Greater than 500 million cases were estimated, were recorded. So that's a massive pandemic that occurred in 1918. HIV and AIDS. Mortality is 35, or greater than 35 million. Let's see, tuberculosis, approximately 1.5 million annually. So that's just yearly, right? 1.5 million people pass away due to tuberculosis. Malaria, 1,600 to date, around 400,000 per year, right? Malaria, it's spread. So what's interesting about some of these diseases is how they're actually transmitted, right? Cholera was through the drinking water, right? Or whatever type of water. Malaria, it's transferred and transmitted through a vector, right? And that vector is the mosquito. And same with like Zika virus and plague, actually. Plague is carried on by fleas, and which then bite rats and things like that. And it spreads out through the population that way. So there's different mechanisms for the transmission. And then last but not least, right, probably the most important right now, most relevant is COVID. And the mortality there is a little more than 5.6 million now, global, right? But there's many other major disease outbreaks to kind of at least list here. And there's been flu pandemics, specifically 2009, H1N1. That's one that I specifically remember when I was in high school. And then Ebola virus in West Africa, which, by the way, right, Ebola virus is still infecting people, right, in West Africa. It's still there. There's many outbreaks that are still occurring. But the one that I worked on was the one in 2014 to 2016. And then there were some other ones, the Zika virus and things like that. But the main thing here is that I wanted to connect it, right? To understand the disease, right, you need to understand much more than what's like the transmission of the disease, but like what's causing the spread of these diseases. And it's what's happening is actually it's a consequence of a lot of habitat destruction, deforestation, development of wildland, losing biodiversity, so you don't have that kind of robustness in your populations. And ultimately, it's that the disease-carrying animals are, in a way, coming closer and closer to human civilization. That's what we're seeing with COVID. That's what we saw with Ebola virus. A lot of these viruses are in the animal population. And every so often, it jumps to the human species, right? Due to, well, living near these animals or whatever, right? So that's a really important thing to think there, right? It's like, why are these diseases occurring? It's because the diseases are occurring in the animals, and we interact with the animals, and then it jumps, right, to the human population. Yeah, so why do we care, right? In 2003, Canada's Advisory Committee for SARS had this wonderful quote. I wanted to say it here. So globalization and public health, right? The world, even now, right, with COVID, we saw it a lot, right? How the world is so interconnected now that, and we're all vulnerable, what's happening in faraway places, right? So a disease outbreak across the world can come here and affect people all over the world, right? So there's a wonderful quote. Yeah, we are constantly a short flight away from a serious epidemic. And in a way, that's what happened with COVID, right? There was an epidemic that occurred, perhaps, right, it's believed to be in China. And then through the interconnected network of flights and transportation, it spread throughout the whole world. Okay, so another question, right, is how does mathematics play a role in this, right? What can mathematicians, computer scientists, or researchers, what do they do when it comes to understanding the dynamics of some type of disease? So I've listed a couple of things here, right? Building testing theories, assessing quantitative questions. That's really important, right? Quantitative questions, right? It's easy to say like the disease is spreading fast, right? Or it's spreading slower than it was. It's really useful to say how fast, right? How many new people are infected each day, or how many new infections occur based on putting one infected individual into a population, right? How many new infections would occur? So the mathematical modeling is really important because it allows us to answer questions from a quantitative perspective, right? Put percents on things, put rates, and that's really important. And also, right, planning, implementing, comparing, evaluating, optimizing various detection, prevention, therapy, and control programs. So a whole bunch of things can be kind of assessed using mathematics. And then 3 and 4 here, right, identifying the trends, making general forecasts. The forecasting for disease dynamics or the spread of the disease is in its infancy. In a way, right, the community would like to have some kind of like a forecasting or reconnaissance program for the spread of diseases and emerging diseases. Almost in a way like the weather, right? How in a way, right? Wednesday, right? It's been forecasted. We're supposed to expect this huge snowstorm, right? And it's something like that, but for diseases would be, yeah, I think that's where the future, a lot of people are putting a lot of resources and time into. The last thing here is early estimation of, right, specific epidemiological thresholds, right? R0 or R0, the basic reproduction number. You probably have heard of that in the news when it comes to COVID. And then the disease burden, right? And disease burden gets defined very differently depending on who you talk to, right? It can be economic, right? The burden on the economy, or healthcare services, things like that. Or you could combine all those things, right? Quantify the disease burden in some sort of way. Yeah, so that's the role of mathematical modeling in disease dynamics, right? It has a lot, right? And in a way, right, this could be applied to not just infectious diseases, but any kind of modeling problem, right? The growth of a cancer, or just the growth of a population, right? The spread of a invasive species, something like that, right? Okay, so last seminar 2 weeks ago, Dr. Johnston, he brought up the compartmental model for a simple spread of a disease through a population. I thought I'd just kind of quickly go through that real fast, just to recall that. But here, right, S, I, and R are the susceptible, infectious, and removed individuals. Removed are, sometimes people call that recovered. In this case, I'll just say removed. And the important threshold, right, for an epidemic to begin is, well, suppose that at time 0, right, I at 0, you have one infectious individual, right, in the population. So think of a whole city, there's no disease there, no COVID, and then you introduce one person with COVID, right? And then you put the condition, you want the rate of change of the infected individuals at time 0 to be positive so that it increases, right? That's how you define, in a way, right, the start of an epidemic, right, mathematically, right, in this model. Well, given that, you can work through some algebra here, right? Plug these conditions into the actual differential equation for the infected individuals, right? And suppose at time 0, right, you have whatever S0, right? S at time 0, that's the number of susceptibles. That would be like the whole city, right? The whole population of the city. You plug it in, right? And at I at 0 is 1. Just plug it into this differential equation. You get this inequality here. And, well, you can rearrange that so that you have some type of fraction, right? Some number greater than 1. That's what we call the basic reproduction number. Beta here is the transmission rate or the contact rate between people, right? The disease. Gamma here would be the recovery rate, right? So infected individuals leave at some rate, gamma, and they enter the recovered or the removed class, right? They're removed from the infection. Yeah, so here's our first quantitative aspect of a disease, right? The basic reproduction number tells us on average how many new infections would occur if you were to introduce one infectious person into an entirely susceptible population, right? So that's an entire city, no disease, throw one person in there with the disease. Basic reproduction number tells you how many new people on average will be infected, right? Quantitative, right? Really important, right? Puts a number on things, right? No longer is it will infect a lot of people, right? It will infect 9 people on average, right? Or something like that. Also, the basic reproduction number tells you some insights, right? How do you eradicate the disease and control it, right? This is a really basic model, so it's only dependent on beta, gamma, and the initial susceptible population. But it tells you that you can reduce R naught below 1, right? Because then that will tell you that if you were to put one person into the population, on average, that person, right, if R naught was greater than 1, or if it was like 5, it would infect 5 people. If it's less than 1, you would expect the disease to die out, right? It wouldn't spread. So how do you control that reproduction number? It tells you, right? How do you make this fraction small, right? How do you make it less than 1? You can increase gamma, the recovery rate, the removal rate, makes sense. Or you can decrease the infectious contact rates, right? The contact rate there, beta. And that will, right, reduce the fraction. Lower the fraction, right? That's probably the word, reduce the fraction. Decrease the effective value of S0, right? So that, right, you probably have heard, right, quarantining people, right? Taking those susceptible people, isolating them, putting them into lockdown, right? That's one way of reducing S0. And I'll just give some examples for, right, it doesn't need to be people, it could be plants, right? A farm, you have some kind of disease spreading through crops. You can reduce and they actually do this in agriculture, right? They go out and they'll just dig up the crops that are infected, cull them, right? Remove them from the population. Yeah. David, do you have a question?
Yeah. Speaking of quarantining, I would assume that would quarantining would decrease beta, which would be the contact rate, and decreasing S naught would be like getting a vaccine.
Right. Yeah. So definitely vaccination will decrease S naught. Yes. Mm-hmm.
Okay.
Yeah. Yeah. Mm-hmm. Yeah. So that's the interesting thing, right? Is the math tells you, right, what needs to be done, and you can find many different ways to reduce that S naught, right? Vaccination programs, some kind of lockdown procedure. Probably quarantine would be for the infected individuals, perhaps, right? Maybe I shouldn't have said that. Social distancing, right? That would be reducing the contact rate, the beta. So it all kind of mathematically makes sense, right? If you're just purely doing this for just math, reducing, making sure that fraction is less than 1, you have those mathematical conditions, right? And they turn out to have real world interpretations. Okay, so I'm glad, David, you mentioned vaccination because that's my next example, right? Suppose you do vaccinate some type of percent of the total population, N. So we vaccinate rho percent of them. Well, then the new susceptible S0 would be N times that 1 minus rho. Plug that into your differential, or plug that into the formula, right, for the basic reproduction number, and you get some kind of critical vaccination percentage so that you can force it, the reproduction number, to be less than 1. And that's where this term herd immunity threshold comes in. And for many diseases, it's different because every disease has a different type of R0, R naught. And it changes, right? R naught for COVID, the Delta variant, Omicron variant has really changed, right, over the last couple of months and year, or last year. So there's another quantitative aspect, right? It tells you how much you need to vaccinate the population, right? And it depends on, well, on that factor, R0, right? The number of new infections that would occur on average from throwing one infected person into a population of susceptibles. Well, you can plot that, right? It's just, it's a function, right? 1 minus 1 over x. You plot it, here's what you get, right? And you can start understanding how fast and how much herd immunity you need to start controlling the spread of diseases. Measles is really high, right? Measles transmits really, really fast. And so for measles, yeah, the herd immunity threshold is quite high, right? 93% or so, right? Really high. Chickenpox, very similar. Smallpox, polio, right? And then COVID, at least this probably is a little out of date right now, but I think the Omicron variant is much higher than what's posted here. But it tells you, right, how much of the population that needs to be vaccinated. So it tells the policymakers, right, how many people they need to make sure that get the vaccine. Right? Okay. So there's another example of just how mathematical models can just quantitatively help us out, right? And that was a very basic model, right? 3 differential equations. We didn't consider hospitalizations. We didn't consider the waning immunity, right, of a vaccine or something like that. Yeah. So just to put everything kind of into perspective here, right, the main point, the motivation here is that the disease in animal populations, I wanted to come back to this because if you go back and look at a lot of the previous epidemics that have occurred recently, Ebola, SARS, COVID-2, right? SARS, MERS, the Middle Eastern Respiratory Syndrome, right? They're all really coming from animal populations. And interestingly, a majority of them come from bat populations. And then they all kind of go up the population ladder here, and then somehow, yeah, it makes it the jump to the human population. So a couple of key facts here is that the actual virus for COVID-19, right, has been kind of circulating in bat communities for 30 to 40 years, right? It's been there, right, for quite a while. It doesn't really harm the bats too much, right? But when it jumps to the population for humans, right, it's, well, we all know, right? It's not very kind to us. So the broader view, right? Many infectious disease outbreaks are linked to animal diseases. Okay. So if there's anything really to take away, right, is from this talk is that the link between animals and disease outbreaks. Okay. Yeah, so now let's jump into kind of some of my previous research projects. Oh, I see there's a question here, Chris. So to get rid of viruses, do we need to get rid of bats, or would some other animal just take up the role of disease? That's a great question, Chris. And I'm sure, right, not all viruses are coming from specifically bats. I'm sure that there are many other viruses, right, that circulate in other animal populations that jump to other animal species. Yeah, great question. Some of my previous research projects have been on, so Ebola virus, 2013 to 2016. So if any of the students are interested in perhaps learning about Ebola virus or Zika virus or any of these, there are, I believe I put the references at the end here for you to check out. But I worked on Ebola virus, so that's spread through bodily fluids. And one of the main mechanisms for the spread of Ebola during the 2013-2016 outbreak was like funeral burials, where what they would do is they would be washing the body before the funeral. The whole community would come together, and Ebola is spread through bodily fluids. So they would be washing the body, and then they'd go back to their separate communities and spread the disease that way. That was one of the main mechanisms. Zika virus, that's spread through a vector, right? That's a vector-borne disease. That's through mosquitoes. And I've also done some kind of retroactive studies in the 1905 plague epidemic in Bombay, which is now modern-day Mumbai. But in that case, in 1905, there was like a really substantial research outpost that was set up there that studied the spread of the plague there. And the research outpost, they wrote this really long document documenting everything, right, collecting data and everything. I mean, modern times now, right, the data collection is amazing, but for 1905, they did really well. I've also worked on some forecasting challenges, right, where you make up a synthetic epidemic that you know how it turns out, and then you challenge other people to kind of forecast what it will look like. And last but not least, I also have done some work with agriculture, so the spread of like a virus through a crop population, specifically like some cereal plants, which is really important for food supply. Let's see, yeah, so just diving a little deeper into some of the previous projects. So Zika virus, what we used there was really just some simple phenomenological models, right? So these are not mechanistic models. They don't model number of susceptibles, infected individuals recovered. They try to more just model the shape of what the data looks like, right? And then can you use that shape of the data, find a model that will kind of try to predict that trend, right? And so I've done some work there with Zika virus. The important thing there, though, is even the simple, really simple models that you've seen before, right, in your differential equation courses, they can be used to estimate reproduction numbers and things like that. And that's really important in the start of an epidemic, right? At the very beginning, you don't have much data to calibrate these really complex models. But if you can use some kind of a 1 parameter or a 2 parameter model, estimate those parameters from the data, interpret it, try to figure out what the reproduction number is, it's really useful, right? So there's kind of like levels of usefulness of the models. Here we were able to find how the reproduction number changes depending on what's called the generation interval. Generation interval, imagine like the time it takes from me becoming infected by someone, and then the time it takes for me to go and infect someone else, that's the generation interval. And there's also something called the serial interval, serial generation or serial interval. The generation interval tries to approximate that. But it makes sense. It's the number of generations that are occurring when it comes to the spread of the disease. Yeah, so we were able to find with Zika, the reproduction number was around about 2.2. And this was early on in, I forget where this data was coming from. It doesn't say in the title either. Yeah. Yeah. Can't remember the name, but in the paper, it's there. Let's see. But the key thing there, right, is we were using a simple model to just forecast how the disease would spread and forecast the number of new cases, right, that might occur. And this is kind of like a retroactive study. And here you see, right, there's a bunch of simulations, right, a bunch of realizations of what could occur, right, from the model. And then the dashed lines here are like the 95% confidence interval, prediction intervals, right, for what's what you can expect. Okay, so that's Zika. That's one of the projects I worked on. The other project that I talked about is the 1905 plague epidemic. In that case, there's a whole document, right? And this is kind of like the first page of that study that was done by that research outpost. And they talk about everything. And they collect data, they map everything out. And kind of like with the John Snow example, they even plot where the cases were registered, where they were finding the cases. And so in this research project, it was more about studying how spatial configurations in a city, how that affects the spread of the disease. Because I don't think I have it here, right? But in Bombay, this is just a part of it, right? This is a fort that was located there. And outside of the fort, there was this big field, right, that kind of isolated the fort from the rest of the city. And so you had this interesting configuration of the city and how that influenced the spread of the disease. So we studied that in one of my previous research projects. Switching real fast to disease agriculture. I've worked with some spread of a virus through a cereal species, a subgroup of viruses called barley yellow dwarf virus. It's called yellow dwarf virus because, well, when it infects the plant, it turns them yellow and then it stunts the growth, right? And so you can see this in specific crops. And so I've put a little photo here of what it looks like. Here's what they look like under the microscope, the actual virus particles. But for this project, I was really interested in, and my collaborators were interested in, how do fertilizers affect the amplification or de-amplification of a virus in a plant? And how does that affect the overall spread in a crop? And so what they did in that case, they went to their lab. I worked with some biologists, and they grew these plants and they infected the plants with this virus. And then they grew the plants under 4 different like nutrient configurations, nutrient treatments. One was a control treatment, like a usual typical fertilizer. The next one was like amplified with nitrogen. Another one was amplified with phosphorus, and then another experiment with both nitrogen and phosphorus. So the idea there was, right, virus particles, right, protein capsid basically, it needs specific types of nutrients, right, to replicate itself. And so do you think there will be an amplification of the replication depending on if there's more nitrogen or more phosphorus, things like that. We've seen things as well with cancer tumors. Phosphorus is like a really vital nutrient for the spread of a tumor, well, specific tumors perhaps, right? Not all tumors. And so this kind of idea of how do nutrients change and influence the spread of disease was a question they were interested in. A lot of the projects, right, are always motivated by some question, right? The math model is used to answer a question, to quantify an answer. So in that case, yeah, we came up with a compartmental model, kind of like the SIR model that I showed before, but V here is virus particles. I is the infected host cells. So this is happening inside the plant, and susceptible host cells. And what we did was, well, we wanted to unite this with resource concentrations of the nutrient. So some type of external resource supply, right? The fertilizer, right? We put in the ground in the plants. Those nutrients were taken up by the plants. That's what this Q is. So Q is the resource concentration within the cells of the host. And then that will influence, right, recruitment of susceptible cells, right, the growth of the plant. It will change, it could change, right, the infectivity, right, of the virus, and the mortality rates of the cells. Has a lot of potential avenues, right, to kind of change how all these dynamics work. This was the model. I just wanted to show you, right? It's a little more heavier than the model that we looked at before, but here are the resources or references if you're interested in checking it out. Let's see, the basic idea was to take an SIR type of compartmental model and just introduce growth functions that were based on the nutrient in the cell, the nutrient concentration. And that's Q in this case. So kind of Q is the nutrient concentration in the cell. It will impact how that cell grows, which impacts how the plant grows. And it will also impact how the virus replicates. And so that's why I highlighted those two there. Otherwise, it's basic mortality of the cells, right? Over a certain period of time, they decay and things like that. And loss from cell growth, right? So there's like a conservation law occurring here with the nutrients. So those are some papers there. The second paper here is more of like a mathematical study. The first one here is kind of building the model, applying it to data, calibrating it, seeing what it tells us. The second paper here is a slightly different model, but it's more mathematical. It's really just a mathematical study of the whole system. Okay, so if you're curious, you can check those out as well. Current research project. So this is what I'm currently working on with Dr. Johnston and Dr. Nelson. We've been working on COVID-19. Dr. Johnston has been working on Virginia data. I've been working on some Michigan data. Here's some of the data here. And what I was kind of, or what we were really interested in was how does this temporary immunity for the vaccine, right, affect the whole dynamics, right? And can we use that to kind of understand some of the more linear growth trends that we're seeing here, right? Is there like a slow increase of susceptibles because vaccinated people are losing their immunity, right? Things like that. So here's another typical SIR. The only thing new here is recovered people return, or infected people recover, they go to the return class. But then over some delay, right, there's a proportion, that same proportion will lose their immunity, right? Because over time it decays, and then they re-enter the susceptible class. And so this is kind of like a simple study or a simple model to characterize some of the dynamics for a fixed immunity period. But what's interesting is that this will produce waves. It produces disease waves. So there's a hot bifurcation occurs and you get periodic solutions. So what I was interested in was taking this simple model here, right? This says here that every individual has a specific number of days that they are immune, right? And that's omega in this case. But it might be different for other people, right? Maybe it's like a distribution, right? The omega, the number of days is distributed in some specific way so that it's not just a specific number every time for everyone. If you wanted to kind of incorporate some kind of distribution for the time it takes to lose immunity, you can write it with some type of what we call like an integral delay or a distributed delay. And that's what's occurring here. And there's actually two types. There's one for vaccinated individuals. So a vaccinated individual, right, is removed at some rate, dV/dt. But then over time, this integral, and here G is the gamma distribution. So it's like the time it takes for something to occur. Those vaccinated individuals return to the susceptible class. So there's like a nice kind of distributed delay. This integral term here, you'll notice that it shows up below in the recovered class. And it's actually mimicking what's going on in that, in this model, right? All I did was just replace this term here with a distributed delay. So it no longer just happens at a specific time, it is distributed. It turns out you can do some really interesting math, right? Convert this into just an ordinary differential equation, system of differential equations, which allows you to solve it really quickly based on very normal, usual methods for solving ODEs. And so here are just some predictions that the model gives based on different immunity durations, right? For Michigan, so this is calibrated to Michigan data. Here, the number of people that are infected. So this is starting around March 2021. You get that peak, and then people start losing immunity and re-entering the susceptible class, and then you start seeing a more gradual linear increase in the cases. And here, I did some predictions based on changing that period. So the baseline case was 242 days, and I switched it from 194 and increased it to 323. And you can see how the dynamics change, right? How the epidemic is different. I mean, so, right, this doesn't account for Omicron or the Delta variant, right? But it's still useful in understanding how at least this disease could be spreading. And actually, when it comes to Delta and Omicron, right, you can kind of interpret the loss of immunity from the vaccine based on having a different variant, right, in the population, right? So the vaccine is less useful against the disease. Yeah, so that's a project I've been working on with Dr. Nelson and Dr. Johnston. And then last but not least, I know I'm kind of pushing my time here. I'm also doing some biological work with a biologist at Oakland University. He's really interested in how climate change affects the disease in an animal. And so what they're working on is what's called thermal performance curves. What they do is they have these labs, right, where they put a frog and they stress the frog under different type of temperature conditions. And then they infect the frog with some type of parasite. And they want to understand how does the temperature affect the performance of the frog, right? Its immune system, its ability to reject the parasite, and also how does the climate, the temperature affect the parasite dynamics, right? Because there's like there's two performance curves going on, and one performance curve might be better than the other, right? And so you might see specific temperature regions where the parasite is more or performs better, right, than what the host can actually do with like its immune system, right? And so how do you kind of put this into a mathematical model? You can borrow some ideas from chemistry, the Sharp-Schofield equation here. It models basically reaction rates from chemistry, but you can apply it to metabolic theory. And you get this nice kind of curve that helps you understand the performance, the metabolism of an animal or a parasite based on temperature. And they've applied this to fruit flies to understand the growth of the wings of a fly, the growth of its thorax, and how the temperature changes, right? And it's exactly what we see, right? Here's the typical curve that you would see from the model. Here's what you see with the data and the curve overlaid on it. But you also see this with specifically what they're working on at Oakland is parasites. So it's a specific type of parasite that infects frogs, and they're able to collect this data, right, in their lab, and they can try to kind of measure the performance of the parasite based on the temperature, right? And they measure the performance in a lot of ways, but one way is like the swimming speed of this parasite in the water. And they videotape it, right? And they can kind of measure it that way. But what's interesting is that you can take these mathematical curves, right? These basic equations, understand how a performance curve for the parasite, understand how the performance curve of the host, and then if you can overlay them, you can see which one outperforms the other based on what temperature it's at. So that's kind of the idea there, understanding how temperature affects the performance, infectivity, things like that. Last but not least, there's even been some more theoretical work where you can now, you have this concept of incorporating temperature into an infectious disease model. You can derive a basic reproduction number like we did at the very beginning, but now it's based on temperature and how temperature changes, how that will change the reproduction number. Okay, so that's, yeah, I think that is it. So that's just a really broad, quick view of some of my research projects that are going on and what I've done before. I don't actually see the reference. I'll update the slide here and I'll put all the references for those, for the students that might be interested in checking out the papers. Yeah, so I guess I'll take questions if there are any. Gus, yep.
I have a question. Did you differentiate between different ages? How do they catch the disease, and or different gender or different race?
Great, yeah. So me specifically, I have not, but you can. There are models out there that will incorporate like the age distribution of a population, the gender. Those models tend to be partial differential equations. So you have now a new variable, the age, which is a continuous variable, but all that can be done as well. Yeah, it's just much harder.
Another question, and I don't want to take too much time, but we live in a world of a lot of misinformation, a lot of information coming across everywhere. Is there a way with your model to detect what is good information versus misinformation? Like we hear in China, the disease is not spreading, they don't have a lot of deaths. Is there a way to say they are suppressing information from us, they suppressing data, or there isn't?
That's another really great question. I've heard of some like metrics that can be used to like measure the spread of information in a population. But I think what you're asking is can you use a model to kind of check to see if there's suppression of that information? And I'm not sure. I want to say that it's not possible or not impossible to come up with some type of model. But I haven't seen any, I don't think.
Yeah, so any other questions for Bruce? We've got about 5 minutes before noon. So if anybody's got.
I'll ask another question. Is there a way in the model to detect whether the disease comes from a plant, an animal, or it was done in the lab?
Well, that's another great question. I mean, what I would say is math models could be a tool for trying to maybe give supporting evidence, right? If either of those cases were true, right? You could use a math model to kind of like, here's the data, we can build that specific mechanism into a model and then say, well, does the model actually fit to the data? And that would provide some kind of evidence, right? But it doesn't necessarily say that that's how it is. Yeah. Mm-hmm. Let's see. I think Paula.
I think Paula had her hand up, and then maybe we'll move on to David.
Right. Sure. Go ahead, Paula.
First of all, great talk, a nice continuation of Dr. Johnston's talk 2 weeks ago. This is the first time actually watching it live. As you all know, I was very overcommitted for January. So yeah, very interesting continuation. Okay, so you mentioned this term, and I was trying to look it up really quickly. Mosquito vector, and then I heard disease-borne vectors. Is this being used? So we understand vector, right? We're in math and computer science as a mathematical object. Is it being used in the same context? And if it is, oh God, okay.
It's slightly different. So it's more like a biological. So like a vector would be like something that spreads the disease, like a mosquito spreads the disease. We call those vector-borne diseases.
So this is, okay, so it's based on the context that it's
yeah.
Okay,
yeah.
Okay, thank you.
Mm-hmm. Right. I think David, you're next.
Yes. One of my questions was related to the severity of COVID-19 presented in the presentation. I noticed that several other pandemics outranked it in terms of mortality and herd immunity threshold, etc. Is this because of, well, some of them, yeah. Is this because the virus currently is less of a threat, or is it because we have taken the correct measures to decrease its threat?
I would say probably both, right? The disease itself is probably, I mean, there is some evidence, right, that the variants have become less lethal, but they spread faster and more contagious. There is some evidence for that. And then there's also evidence for what you said as well. And I'd also say that the technology probably plays a role nowadays. Especially when it came to like lockdown and then people needing to get groceries and order things online. You have this new type of technology that allows you to be kind of isolated from other people, right? So that you don't need to spread this disease.
Isolated but still connected.
Right, yeah, yep. Which, right, in 1918, right, that pandemic there was, right, even communication was hard probably to communicate to a city, right? The spread of a disease or something like that. There's no Twitter or anything right back then.
Yeah. So yeah, it is the resources that we have available to us to deal with. Yeah.
Right. Yep.
All right, so we are right at noon. So I think this is a good time to thank Professor Pell for a wonderful talk, a really great introduction to what he's been working on, really throughout much of his professional career and up to date, and hopefully maybe aided by some of you in the future as well. So we will meet again here. Paula, who was able to log in, and we've seen her. She will be giving the talk on the 15th of February, 2 weeks from today. So I'll thank everybody for signing in and joining us, and we'll see everyone in 2 weeks. And also, Bruce, if you could send those slides over to me, I can post those as well.
Will do.
All right.
Thank you. Great talk.
Yeah. Thanks,
Thank you,
everyone.
everyone.
Thank you.